brighter than the sun. Yeah, the bonnie boat was one as we sailed into the mystic. Oh, I can now hear the sailors cry, smell the sea and feel the sky. Let your soul and spirit fly into the mystic.
Okay, that was Van Morrison's Into the Mystic, and chose that song because it's a great accompaniment to our first lecture, and really to the whole course. Yeah, Into the Mystic. The idea that the things that we're going to be talking about in this class are mundane miracles, right? They're the stuff of it's miraculous what we can do with our physiological structures, and we go to movies and we watch Iron Man and Thor and all that stuff, and we think, wow, wouldn't that be great if we were able to have this magical hammer or whatever. But we kind of overlook the miraculous things that we're capable of and the miracles that we experience every single day, right? That happen because of our natural environment and our natural physiological processes. Okay, so today we're talking about neurons. This is the first half of the lecture for chapter 1. And the purpose of this lecture and the chapter is to introduce folks to the basic structure and function of neurons. Yeah. Want you guys to become familiar with the anatomical structure of neurons as well as the function. Yeah. We're going to talk about how neurons communicate by electrochemical signals, and we'll break that down into what is the electrical process, what is the chemical process. Neurons are capable of processing our sensory information, producing complex behaviors, and everything we know or experience or think or dream is all thanks to the functioning of these individual cells. Yeah. So let's take a look at an overview. Okay, so in this lecture, we're going to start with the history of neurons as cells, talk about Ramon Cajal and his discovery. Then we'll spend the bulk of it on the structure and function of neurons, on the structure of the synapse, and the various types of cells in the brain. There's different kinds of neurons. There's different kinds of glia, which are the support cells for the brain. Yeah. In the second half of the lecture, we'll talk about the communication within neurons, which is the action potential, the electrical pulse that travels down the axon. We're also going to talk about communication between neurons, which is the chemical signal that gets released from the axon and connects to the next neuron via the dendrites. And put those two processes together, and you have what's called neurotransmission. And this is how we function and communicate and get everything done. Yeah. Okay. So to start off with, we've got the history of neurons as cells. For a long time, we didn't even understand that cells exist, right? We knew organs and that sort of thing, but it took until we had the ability to use a microscope and get really small, detailed images that we were able to see that we're comprised of all of these little individual units, right? And so same thing goes for the brain. The person we want to talk about is Ramon Cajal. Yeah, and the reason for that is because he was the man who identified that we don't have a whole bunch of sort of strings of organ in our brain. Yeah, what it was all of these discrete little units, which are called neurons. Yeah, and so this was a huge advance. Yeah, and he was able to do this because he used what's called a Golgi stain, yeah, to stain brain tissue. And that Golgi stain works really well in two ways. One is that it does stain the neural tissue without the other tissue. But another is it only stains about 1 to 2% of that tissue, which is great because then, if you get 100% of the tissue, it becomes, almost a visual, white noise. Yeah. But if it's only 1 or 2% of the tissue, then all the other stuff kind of fades into the background, and we can see individual cells, right? And that's what we have here. This is a drawing from Ramon Cajal, where he's describing or trying to illustrate dendrites, yeah, and that's what all this complex stuff is at the top of the picture. These are all dendrites, and so that's the incoming area, right, for the neuron. And then we also see these sort of long processes that extend down towards the bottom of the image, and those would be the axons, yeah. Okay, so but we'll talk about what those are. What do we mean? What do I mean when I say axons and that kind of thing? But what matters here is that Ramon Cajal is the guy who went against the prevailing theories of the time that said that the brain is an interconnected matrix. Yeah, he identified that we've got individual cells that communicate somehow. He didn't understand the neurotransmitter process and that sort of thing. But he was able to show that our brain is made up of discrete units, not all the same thing. Yeah, and he also basically identified that neurons are the structural or functional units of the nervous system. When you break down the nervous system, the smallest unit you can get to is the neuron. Yeah. Okay, so let's back up a little bit and talk about the general overview of neurons. Yeah. So neurons are what comprise our central nervous system. Yeah. They produce all of our consciousness, all of the functions in our brain, everything that our sensory systems do. It's all thanks to the function of neurons. Yeah. But neurons aren't the only thing that we have in our brains. We also have things called non-neural cells, yeah, and those we call glia. And these cells do different functions to support and protect neurons and their processing of information. Yeah. So we have different glia that provide an additional barrier around the capillaries in the brain called the blood-brain barrier, which we're going to talk about. We have others that help to provide a myelin sheath around axons and speed neurotransmission. Others that sort of work like macrophages and pick up waste and dead tissue and that sort of thing. Yeah. Okay. Right. So the neuron is a cell that is specialized for communication. Right. That's what it does. It does not digest food. It does not help us to go to the bathroom or whatever. It is specialized for communication. And so everything that we think, everything that we feel, everything that we are, and everything that we do depends on these microscopic mechanisms of monumental complexity. Yeah, pardon the pun. But although there are exceptions, these neurons in general share very similar structures and functions that are just a little bit different from each other, but make sense when you look at them in comparison. Yeah. So real quick before we get into the image, talk about what these various parts of the brain cell are. We've got dendrites that take in information, right? Dendrites are on the postsynaptic membrane, or actually they comprise the postsynaptic membrane, and these extend from the cell body, and this is where information comes in. So these are the main input unit of the neuron. Yeah. The cell body or soma is what holds the DNA, right? It contains the nucleus for the cell. It contains a whole bunch of support structures like mitochondria and Golgi complexes and all that kind of stuff, right? So it allows, the cell body is where all the things are done to keep the cell healthy and alive, are completed. The axon is sort of like the roots. Yeah, if the dendrites are the branches of a tree, and the cell body is the trunk, then the axon would be the roots. Yeah, and it's typically one major line, one major. Well, we call it a transmission cable. Yeah, and this axon is where the action potential travels, and this is the electrical pulse that travels down the neuron to get to the end of it, which is the presynaptic terminal. At the presynaptic terminal, vesicles are activated by that action potential and bind to the membrane of the presynaptic terminal and release neurotransmitter molecules into the synapse. Yeah. And what are vesicles? Vesicles are like purses. They're like backpacks. They're just storage transport structures for neurotransmitters. Yeah. Vesicles get filled with neurotransmitters up in the soma, and then they travel down the axon and wait at the end at the presynaptic terminals for an action potential. Yeah. When they bind with the presynaptic membrane and they release their neurotransmitters, those neurotransmitters float into a gap. I'm sorry, between this neuron and the next one. That's called the synapse. Yeah. So a synapse is basically the presynaptic membrane of one neuron and the postsynaptic membrane of the next neuron, and there's a small gap between the two. Okay. Let's take a look at some images. Okay. So here we have, this is from your textbook, right? And I'm going from the left side of the image to the right side. Okay. Top left side, we've got the dendrites. These are the branches of the tree. This is where the incoming information comes. Yeah. Next thing that you've got labeled there is the cell body or soma. Yeah, and that's what contains the nucleus and contains things like mitochondria, Golgi complexes, blah 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 blah. We don't need to get into the whole DNA, RNA transcription stuff. Just know that that's sort of the heart, right? That's what keeps the cell alive is the soma and the nucleus. Yeah. So projecting off of the cell body is a long sort of tube structure that is called an axon. Yeah. And typically we have many dendrites and usually only one axon. Yeah. So you'll notice that this is a long projection and it's covered with these see-through little rectangles of stuff, right? So those rectangles are what are called the myelin sheath, and that's really basically lipid coating, yeah, over the axon. And what that does is it allows for the electrical signal to travel much faster than it would without. Yeah, and we'll talk about why. It's called saltatory conduction, but we'll talk about why. Those nodes of Ranvier, those are the gaps in between the myelin sheath. And so that's important because that's why it travels so much faster, because all it has to do is happen in the nodes of Ranvier rather than throughout the entire axon surface. Yeah. So it sort of skips, it jumps from one node to the next along the myelin sheath, and that makes it much more rapid. Yeah, because it doesn't have to travel through the whole thing. It sort of like teleports from one node to the next. Yeah. Once the action potential reaches the end of the axon, then that's where we've got those terminal projections and the presynaptic membrane. Yeah. And you can see it's just a tiny little gap between the axon of one neuron and the dendrites of another. Yeah. And that's where the chemical process happens. Yeah. Let's take a look at another image. Okay, I like to have a couple of different images when I can, because for me that really helps me to understand things. If I see just one image, then I kind of get stuck. Yeah, but if I have a couple of different images, then I get the sense that I get a better sense for what I'm looking at. So this isn't in your textbook, but I want you folks to be able to have more than just one image to understand what the neuron looks like. So here, top left, we have the cell body. Inside that is the nucleus. All around that cell body or the soma are the dendrites, right, that receive the signals. We've got one long tube projection that is the axon. It's covered with the myelin sheath. Every gap in between that myelin sheath is called a node of Ranvier. At the end of the axon, we've got your terminal branches, your terminal button, and this is where the synaptic vessels bind with the membrane, release neurotransmitters into the synapse. And let's take a look at the synapse. So the image on the left-hand side you have in your textbook. The image on the right-hand side you don't, right? The right-hand side, this is an actual electron microscope image of a synapse. Yeah. So we'll use the terms on the left-hand side of the screen and just go over it real quick. The presynaptic terminal is at the end of the axon, right? That's at the top of our drawing here on the left-hand side. You see these white circles with these blue dots inside of them. Those are called vesicles. The idea is those are the backpacks that contain the neurotransmitter molecules, right? And they float in the presynaptic terminal button, and they wait until they're activated. They're activated by an action potential. Yeah, that electrical pulse hits this presynaptic terminal, and it activates those vesicles to go and bind with the membrane of the presynaptic terminal. Yeah, they don't go outside the presynaptic terminal. They actually become part of that membrane and open up like a zipper. Yeah, and release their neurotransmitters into that little gap. It's called the synaptic gap, or sometimes the synaptic cleft. Right. So, why do they do that? Because those neurotransmitters, pardon me, are communicating information to the next neuron. The next neuron is basically the dendrite, right? And on a dendrite, we have receptors, which are basically protein molecules, typically, right? And what those do are they work like your house door, your door lock. Yeah, you need a key that has the exact right structure to fit into that receptor and cause it to activate, just like your key needs to be exactly correct to fit into your door lock and to open up the door. Yeah. So that once that process happens, basically it changes the electrical potential of that neuron, right? And if it changes enough, it can cause another action potential to fire in that next neuron. Yeah. Okay, so uh, let's look on the right-hand side here. Here is an actual image of what a synapse looks like in the brain. And you can see this is, again, like I said, this is an electron microscope, and it's still, we still would like to be able to blow it up even further. Yeah. So on the very top of that picture image, you've got this red sort of blob. Yeah. And that red blob is the presynaptic terminal button of one neuron. Yeah. Inside that red blob, you see a big black circle or oval. Yeah. That big black oval is not a vesicle. That's a mitochondrion. Yeah. The vesicles are the tiny little purple dots that are surrounding that black oval. Yeah. And you'll notice that those vesicles, those tiny little purple dots, are kind of building up in two different areas down at the bottom of the red blob. Yeah, at the bottom of the presynaptic terminal button. The reason for that is because that's where the gap is. That's where the synapse is. Yeah. So those vesicles will bind with the membrane and release their neurotransmitter molecules that are those little purple dots are filled with these molecules. Yeah. They open up, they release the molecules into those two areas that have kind of a green stain to them. Yeah. And that green stain, that's because that's where those receptor protein molecules are. They float across the gap, they bind with the receptors, and they cause an activation. Something else I want to point out to you is this yellow part, just this top part that's right underneath the red, the postsynaptic, or the presynaptic terminal, is a dendritic spine. Yeah. You'll notice that there's a big swath of yellow underneath the dendritic spine. And what that is actually the dendrite itself. Yeah. And what we're looking at is a tiny projection off of the dendrite that is specific to that other neuron. Yeah. And takes in information from there. So this one dendrite can have 1,000 dendritic spines along it. Yeah. With 1,000 different synapses and connections and that sort of thing. Yeah. So you can imagine when we're talking about that's just one cell. Yeah. One neuron's dendrites. Yeah. That this is it's fair enough to say that the brain is the most complex system that man has ever come across, that we've ever tried to understand. And we're really just at the basic beginnings of understanding what we're doing. Still, though, in the past 20 years, in the time since I've been a student and have become a teacher, we've made amazing strides. Yeah, I never saw an image like this when I was a student. We did not have them. Yeah, but we're getting better, and the benefit is the more knowledge we have, and the better the understanding we have, the better we're able to make things work, yeah, and fix things when they break, hopefully. So we've talked about the history of neurons as cells, Raymond Cajal. We've talked about the basic general structure of the neuron and the general structure of the synapse. Now we can talk about the different types of cells in the brain and the functional and structural classification of neurons, and then we'll talk about glial cells, and that'll do it for this lecture. All right, here we have a basic slide of the three different kinds of functional classifications we have for neurons. Okay, we have incoming neurons that are called sensory neurons, and that's what we have in the blue up at the top of the image, right? The information, hits one of our sensory systems, activates our sensory neurons, and they carry the information into our brain. We also have outgoing neurons that carry information from our brain to the rest of our body, and those are called motor neurons. Yeah, which makes sense if we need to make our legs move so we can walk or whatever. Our brain has a pathway basically where it sends impulses down to the muscles in our legs and causes our body to then move the way that we hope or want it to move, right? But there's also an interneuron. Interneurons are the middleman, right? Interneurons are basically what process all this information and allow for the transfer of something going from a light impulse into a recognized image that we see. That's a car coming. I better not walk across the street or whatever, that sort of thing, and then sends the information to our body that says, do not then step into the street, right? So interneurons basically process that sensory input into meaningful representations. They also help us to plan an appropriate behavioral response and then connect to the motor neurons that execute those behavioral plans. Yeah, so three different functions, right? So then we can talk about the structural classification of neurons. I don't want you guys to worry too much about this. Just the idea that we have different, neurons can take different shapes. The most common one is the multipolar neuron. That's in the center there. And these are what communicate sensory and motor information in the brain. Yeah. They have one axon, they have many dendrites, and it allows them to take in a whole bunch of information from various sources. And then, of course, their axon splits up at the very end there. But we've also got bipolar neurons and unipolar neurons, and these are basically different structures. The bipolar neuron is one that we would find in, for example, in our sensory system in the eye, for example. Yeah, they help with light perception. They have one axon, one dendrite, and their job is to acquire and pass information on. Unipolar neurons are also structured in a way that allows them to relay information forward. Yeah, and so we have in general, when you think of neurons, you're thinking of multipolar neurons, but we also have bipolar neurons and unipolar neurons. So there are different shapes, right, of neurons. But I don't want you to focus as much on that as I do on the functional classification, right, on the difference between a motor neuron and a sensory neuron and that sort of thing. All right, now we get to talk about all the fun support systems that the neurons have. Yeah, and so in general, when you're talking about a non-neural brain cell, you're talking about something that falls under the umbrella of glial cells, right? And so as you can see here, we've got microglial cells, astrocytes, oligodendrocytes, yeah, various different kinds, yeah. So let's take a look at them one at a time. I don't know if it says this in your textbook, but glia comes from the Greek word for glue. Yeah, and this is because when early neuroanatomists were looking at the brain, they thought that these cells were basically structural support. Yeah, that they acted like glue that held everything together, which isn't actually true. But you'll see why they thought that they acted like glue. So let's look at the oligodendroglia. cells. Yeah, these cells provide myelin in the central nervous system and in the peripheral nervous system. Oligodendroglial cells are what speed transmission along the axon of a myelinated axon. Yeah. So we can see, as you can see, the oligodendrocytes are sort of the aquamarine colored cells we have there, and they have projections that reach out from the cell body and wrap around and the axons of different neurons. Yeah, and that's what provides the myelin sheath and speeds the transmission of information. Yeah. So so they see these cells that are reached out and wrapped around other cells, so they assume, okay, so this is to sort of hold things together, right? That makes kind of sense. And then they see other cells. They see microglia, right? Microglia, top left-hand corner of the image here, it looks kind of like a Brillo pad, yeah? And it functions like a microphage in the immune system. Its job is to get rid of substances that don't belong there, right? So it can be dead tissue, it can be something that got through the blood-brain barrier that doesn't belong. It could be an infectious agent, that sort of thing. But it's usually waste. It's usually dead tissue, dead something or other that it just basically gets rid of, right? So it's a little factory for breaking things down. Yeah. Then we have astrocytes underneath the microglial cell, right? Astro means star in Greek, and the reason for that is because when you look at these cells, they do kind of resemble stars a little bit. So what the astrocytes do is they help us to maintain chemical balances in the brain. They remove waste in the brain, and they also perform reparative functions. Yeah, but most important is that, as you can see, they also stick out projections, right? And many of these projections go to the capillaries inside the brain. And what they do is they help to sheath the capillary, yeah, and prevent molecules that are inside the capillary from getting out when they're inside the brain. Okay, let's take a look at that. Okay, so here's a good image of the blood-brain barrier. We have capillaries throughout our body that carry blood cells, uh, and they're basically the same everywhere in our body except for in our brain. Yeah. So this image on the left-hand side is a nice little drawing of a capillary in the brain. On the right-hand side, it's a cross section. It's as if if you cut the capillary and you're looking down the center of it, right? So in the body, our capillaries have little gaps in them that allow molecules and blood cells to go in and out. Yeah. So they aren't super restrictive there. But when the capillaries get into the brain, their the wall of the capillary itself becomes much more dense. Yeah. So those gaps are much smaller, and that means that for anything to get out of the capillary, it has to be a specific structure and it has to be a specific size. Because not only does it have to get through the capillary now, it also has to get through that glial cell wrapping that you have around the capillary, right? And as you can see here on the left-hand side, the astrocyte, right, is a glial cell. It sticks out these projections that wrap around the capillaries in the brain and prevent many different molecules from getting out. Yeah. However, you're probably thinking to yourself, okay, but we know that people take drugs, for example, and those drugs then affect the brain. So how does that work? Well, like I said, most molecules can't get through the blood-brain barrier, but every psychoactive drug that you know of, LSD, psilocybin, THC, alcohol, any drug that affects the brain is able to get through this blood-brain barrier, right? And that's because of the size and structure of the molecule, right? The more fat-like or lipid-like a molecule is, the easier it's going to be for it to go through the capillary membrane and through the glial cell membrane and out into the brain. Yeah. If you want to know more about that, we'll talk more about that in Psychology 405, Biological Correlates of Substance Abuse. But know, that we've got, the myelin sheath on axons and then we've got the blood-brain barrier on brain capillaries. Yeah. Okay, so that's going to do it for the first half of the lecture. I wanted to give you a little bit of guidance on the discussion that's due this Friday. Yeah. So for this Friday, you need to read the article. I think it's only like 3 or 4 pages. Top Mysteries of the Mind, Insights from the Default Space Model of Consciousness. Okay, this is posted on the website. And it's in the discussion articles folder. When you click on the syllabus, textbook, and lectures page, it'll open up a bunch of folders, and one of those is discussion articles, and you'll see this in there. So what you have to do is read that article. I think it's really interesting, but all it does is really introduce these various mysteries of neuroscience, right? Questions that have not been answered yet. So what I want you to do is choose one of those questions that really stands out to you. Explain what that issue is. Why is it important? Why do you find it interesting? Make sure that you give an accurate description of what that mystery is, right? And you get that from the article, yeah. And then give me your take on it, yeah. Why did you choose this one? And what do you think is so interesting about it, and that sort of thing. So here are the questions, the questions that you're going to see in the article. What is the relationship between subjective experience and the physical world, right? Our subjective experience is the fact that you and I can go see a movie, and the two of us can have very different experiences at that movie. Or, for example, we could be walking down the street and see a minor fender bender and have two very different ideas about who caused it, what happened first, all that other stuff. So there is a physical world in which objective things occur, but all of our experience is subjective. It's based on our own personal perceptual makeup. Yeah. Next question is, how do we so quickly process and interpret the external world? Yeah. We have these 5 sensory systems, and they function incredibly rapidly. Yeah. Not only do they bring in, information about light and vibration and pressure and heat and smell and all that other stuff, but it all gets molded together into one seamless experience. And that's our third question. How do all of our sensations unify into this one conscious experience that we have? Where it's not a sense of I hear, there's a sound, so I have to focus on my sound system, or I have to focus on my visual system, or now I have to walk, so I have to focus on my physical system, on my legs. It all just happens, right? We don't have to think about it. We don't have to divide it. We say, oh, I can hear a bird chirping, and I can taste, or I can hear a chicken crowing, right? And it just happens for us, yeah. Fourth question is, why do we sleep? Why do we spend 1/3 of our lives unconscious? Yeah. And there's different kinds of sleep, right? And there's we know that different animals sleep in different ways. For example, it seems that dolphins sleep by shutting off half of their brain, right? One half goes to sleep, the other half is still in a waking state, yeah, and then they flip over, yeah, and the other one goes to sleep and the other one's waking. And then finally, how and by what mechanisms can emotions be regulated? Yeah, and that's a big one, especially for parents who are probably thinking, yeah, I would really like to be able to identify the mechanisms by which my child's emotions can be regulated. Yeah, I'd like them to not be so, whatever it is, right? Loud, or help them to be happy, healthy, not angry, not anxious, all that other good stuff, right? So these are the 5 questions you're going to read about. Read the article and then choose one of those. Explain what that mystery is, and tell me why you think it's important and why you find it interesting. And so that'll do it. That's the end of this lecture. Next, you want to get working on that discussion topic. You've also got Neurons Part 2 lecture to do. When you do your discussion topic, you're encouraged to respond to other students' questions and things like that. There's no rule against that. I don't give extra points for it, but it demonstrates an engagement with the class that can be very beneficial when there's an issue that I have to, where there's sort of he said, she said, or whatever. It's a good idea to demonstrate that you're engaged in the class. Okay. Also, don't forget this week your quiz 1. That's on just this chapter on neurons only. It's 20 questions, multiple choice. It's going to be open from 8 a.m. to 11:55 p.m. on Friday. Okay. Make sure that you're familiar with the test taking program. Make sure that you save each question when you answer it. Yeah. And then contact me with questions or concerns. Please send me an email if you want to set up a phone interview or that sort of thing. I'm happy to do that as well. Okay. All right. Until the next lecture. Thank you very much for hanging out with me, and a hui ho.